Hello, curious minds, and welcome to our deep dive into the incredible chemistry of DNA and genetics. Today, we're going to journey from the very basics of what DNA is to how it's made, why it's made, and how it powers the machinery of life as we know it. Whether you're a student, a science enthusiast, or just someone fascinated by the magic of life, this exploration is crafted just for you. So sit back, grab a cup of coffee, and let's unravel the chemical code that makes you. Part 1. What is DNA? DNA, short for deoxyribonucleic acid, is like the instruction manual for life. It tells cells what to do, how to do it, and when to do it. Everything from the color of your eyes to how your stomach digests food is influenced by this amazing molecule. What's DNA made of? DNA is made of tiny building blocks called nucleotides, and each nucleotide has three parts. A phosphate group, think of it as the connector. A sugar called deoxyribose, a base, one of four chemical letters. A, adenine, T, thymine, C, cytosine, or G, guanine. The bases are like letters in a code, and the order of these letters is what makes every living thing unique. The famous double helix, imagine a twisted ladder. That's the shape of DNA, called a double helix. The sides of the ladder are made of sugar and phosphate, and the steps are made by pairing the bases. A always pairs with T, C always pairs with G. These base pairs are held together by hydrogen bonds, kind of like gentle Velcro that keeps the strands together. Why is DNA so stable? Even though DNA is tiny and delicate, it's incredibly stable. The spiral structure and the way the bases stack on top of each other help protect it. It's like a well-designed spiral staircase that doesn't wobble. DNA versus RNA. What's the difference? DNA has a sibling called RNA, ribonucleic acid. Think of DNA as the library where all instructions are stored, and RNA as the photocopy of a specific recipe that a chef, your cell, uses to cook up proteins. RNA is single-stranded, uses a different sugar, ribose, and swaps out thymine, T, for uracil, U. DNA in your cells. Believe it or not, every cell in your body, except red blood cells, has a complete copy of your DNA. That's about 2 meters, 6.5 feet of DNA in each cell, tightly packed into the nucleus using proteins called histones. The DNA wraps around histones to form structures called nucleosomes, which fold further into chromatin and then into chromosomes. Humans have 46 chromosomes, 23 pairs, with about 3 billion base pairs of DNA in total. That's a lot of code. More than just genes only. About 1 to 2% of your DNA actually codes for proteins. Those are your genes. The rest? Scientists used to call it junk DNA, but now we know it plays important roles in regulating genes, protecting DNA, and helping chromosomes do their job. A living code. You can think of DNA as a super advanced biological computer code. The A, T, C, and G letters are instructions for making proteins, the little machines that do all the work in your body. And just like computer code needs to be written and read properly, so does DNA. We'll talk more about how the body reads this code in Part 3.
Part 2. How is DNA made? Now that we know what DNA is, let's talk about how it's copied. Every time your body makes new cells, which it does trillions of times a day, it needs to copy your DNA perfectly. This process is called DNA replication. Copying the code DNA is copied using a method called the semi-conservative model. This means each new DNA molecule keeps one of the original strands and adds one new strand. It's like copying an old recipe by using half the original page and rewriting the other half. Where it all starts. Copying starts at special places on the DNA called origins of replication. In humans, there are thousands of these. DNA likes to open up in places where A and T are paired. They're easier to pull apart than C and G, unzipping the DNA. An enzyme called helicase acts like a zipper puller. It unzips the two strands of the DNA ladder. Then, single-strand binding proteins come in and hold the strands apart so they don't stick back together. Getting ready to copy. Another enzyme called primase lays down a short starting point called a primer. This is like placing a bookmark to mark where to begin copying. The master copier, DNA polymerase. Here comes the star, DNA polymerase. It adds new nucleotides to build the new DNA strand, using the original strand as a guide. It can only add to an existing chain. That's why it needs the primer. The leading strand gets copied smoothly in one go. The lagging strand is copied in small pieces called Okazaki fragments, which are later joined together by another enzyme called DNA ligase. Built-in proofreading DNA polymerase isn't just fast, it's careful. It has a proofreading ability. If it makes a mistake, it catches it and fixes it. Thanks to this, DNA is copied with an error rate of about 1 in 10 million. What about the ends? Here's a challenge. DNA polymerase can't fully copy the very ends of DNA strands. Over time, this could cause important DNA to be lost. But nature has a solution: telomeres, which are protective caps at the ends of DNA. There's also an enzyme called telomerase that helps rebuild them, especially in stem cells and cancer cells. A quick look at the big picture: DNA replication happens during a specific part of the cell's life called the S phase. With the help of dozens of proteins, the entire human genome can be copied in just a few hours. It's like photocopying an entire library, with almost no typos, every time your body needs a new cell. And there you have it. DNA isn't just a static molecule. It's alive with activity, constantly being read, copied, and used to build you. In Part 3, we'll explore how your body reads the DNA code and turns it into action by making proteins that do everything from growing hair to building muscles. Stay tuned.
Part 3. How DNA makes proteins. In Part 3, we're going to uncover the real magic: how the instructions in your DNA become living, working parts of your body. It all happens through a process called gene expression, which has two main steps: transcription and translation. Let's break this down in everyday language. What are proteins and why are they important? Proteins are like tiny machines or tools that run everything in your body. They help you digest food, build muscles, fight infections, and so much more. And guess what? Every protein starts with a gene, a stretch of DNA that gives specific instructions for making it. So how does the cell read the DNA and build a protein? Let's find out. Step 1. Transcription, writing down the recipe. Think of transcription as making a handwritten copy of a recipe from a cookbook. The cell finds the gene it needs in the DNA. An enzyme called RNA polymerase unzips that part of the DNA and starts building a single-stranded copy. This is messenger RNA or mRNA. Instead of thymine, T, RNA uses uracil, U. So if the DNA has A, the RNA gets U. This mRNA copy is like a recipe card. It's shorter, mobile, and ready to go. When the mRNA is done, it leaves the nucleus, the library, and heads to the ribosome, the kitchen, in the cytoplasm. Step 2. Translation, making the protein. Now we're in the kitchen, the ribosome, where proteins are made. The ribosome reads the mRNA 3 letters at a time. Each 3-letter code is called a codon. Each codon tells the ribosome which amino acid to add. Little helper molecules called tRNA transfer RNA bring the correct amino acid to the ribosome. It's like reading a recipe line by line and adding ingredients one by one. The amino acids link together to form a polypeptide chain, which folds into a specific shape. That's your protein. How does the cell know where to start and stop? The mRNA has a start codon, AUG, to begin the recipe. There are also stop codons that tell the ribosome that's the end of this protein. Final touches: protein folding and modifications. Once the protein is made, it often needs to be folded into a 3-dimensional shape to work properly. Some proteins also get extra parts added or are sent to specific places in the cell. It's like finishing a dish and deciding whether to plate it, package it, or put it in the fridge. Why it matters. The way your DNA gets read and turned into proteins is the foundation of biology. Every action in your body, from healing a wound to thinking a thought, depends on this chemistry. When this process works well, everything runs smoothly. When it doesn't, like in genetic diseases or cancer, things can go very wrong. That's why understanding it is not just cool, it's incredibly important. And that wraps up Part 3. Now you know how the silent code in your DNA becomes real, living proteins. In the next part, we'll explore how genes are inherited, what makes you different from everyone else, and how modern genetics is changing medicine and the future. Don't miss it.
Part 4, Inheritance, Genetic Differences, and the Future of Genetics. We've learned what DNA is, how it's made, and how it helps build proteins. Now let's talk about how DNA is passed on from one generation to the next, what makes each of us unique, and how science is using this knowledge to shape the future. How traits are passed down. Let's start with a big question. Why do you have your mom's eyes or your dad's nose? It all comes down to inheritance. You inherit your DNA, your genetic instructions, from your parents. Half comes from your mom, half from your dad. That's why you're a unique combination of both. Your 46 chromosomes are actually 23 pairs. Each pair includes one chromosome from your mom and one from your dad. These chromosomes carry genes, which are the instructions for traits like height, blood type, and eye color. Each gene can come in different versions called alleles. For example, there might be a gene for eye color with a blue version and a brown version. If you inherit the blue allele from both parents, you'll likely have blue eyes. If you get one blue and one brown, the brown might dominate, depending on how the alleles interact. Dominant and recessive genes can be dominant or recessive. A dominant gene only needs one copy to show up in your traits. A recessive gene needs both copies to be the same to show up. Think of it like volume control. A dominant allele is like turning the music up so loud you can't hear the other. A recessive allele is like a soft background tune. You only hear it when it's playing alone. What makes you unique? Even though humans share more than 99.9% of their DNA, the tiny differences, just 0.1%, make us unique. These differences are called genetic variations, and they're why people have different hair textures, immune responses, or even taste preferences. One common type of variation is a SNP, pronounced SNP, short for single nucleotide polymorphism. It's when a single letter in your DNA is different. For example, most people might have ATG, you might have ACG. These tiny changes can influence how your body reacts to foods, medicines, or diseases. Genetic disorders, when the code has errors. Sometimes changes in DNA can cause problems. These are called mutations, and while many are harmless or even helpful, some can lead to genetic disorders. For example, cystic fibrosis is caused by a mutation in a gene that affects lung and digestive function. Sickle cell anemia changes the shape of red blood cells. Some forms of cancer are linked to mutations in genes that control cell growth. Some of these mutations are inherited. Others happen during your life, especially if your DNA is exposed to harmful chemicals, radiation, or errors during replication. Epigenetics, turning genes on and off. Here's where it gets really cool. Just having a gene doesn't mean it's always active. There's a field of science called epigenetics that studies how genes are turned on or off, kind of like light switches. Your environment, lifestyle, and experiences can all influence gene activity. For example, stress can turn on genes linked to inflammation. Exercise can activate genes related to energy use and brain health. Even what your grandparents ate or experienced might affect your DNA activity today. That's how powerful epigenetics is. Genetic testing, what can we learn? Thanks to advances in science, we can now read your DNA using genetic tests. These tests can trace your ancestry and heritage, identify your risk for certain diseases, show how you might respond to medications. Doctors can use this information for personalized medicine. Treatments tailored to your DNA. It's like having a custom-made health plan based on your unique code. CRISPR and gene editing, changing the future. Imagine if we could fix bad genes. That's the promise of CRISPR, a powerful tool that lets scientists cut and edit DNA. With CRISPR, we might be able to cure genetic diseases, make crops more nutritious, prevent the spread of inherited disorders. It's exciting, but also raises important questions. Should we edit the genes of unborn babies? How do we make sure it's safe and fair? These are the ethical puzzles of the future. The big picture DNA isn't just a molecule, it's a living, changing story passed down from your ancestors, shaped by your environment, and influencing your health, identity, and future. Understanding genetics helps us appreciate how connected we all are and opens up possibilities for a healthier world. And that wraps up part 4. Now you know how DNA is inherited, what makes each person unique, and how the future of genetics could transform our lives. Part 5, how genetics shapes health, disease, and medicine. Welcome to part 5. Now that we understand how DNA works, is inherited, and how it varies between people, it's time to explore how it affects our health. This part dives into how our genes influence our risk for disease, how they interact with our environment, and how this knowledge is transforming medicine today. Your genes and your health. Have you ever wondered why some people seem to get sick more often than others? Or why some can eat anything without gaining weight while others struggle? Part of the answer lies in your genes. Genes provide instructions for building proteins that keep your body running. But if those instructions are a little different, or if something goes wrong during copying, the resulting proteins might not work properly. That's where health problems can begin. Here are some ways genetics impacts your health. Inherited conditions. Some diseases are passed directly from parents to children. These are called genetic disorders. Example, Huntington's disease is caused by a single faulty gene. If one of your parents has the gene, you have a 50% chance of inheriting it. Predisposition to disease. You might inherit a higher risk, not a guarantee, of developing certain diseases. Example, BRCA1 and BRCA2 genes, if mutated, greatly increase your chance of breast and ovarian cancer. Gene-environment interactions. Your genes and your lifestyle work together. Smoking, diet, stress, and pollution can trigger health problems in people with certain genetic vulnerabilities. Common diseases with genetic links. Let's look at some common conditions where genetics plays a role. Heart disease. Some people inherit genes that affect how their bodies handle cholesterol. These genes can make you more likely to develop clogged arteries, even with a healthy lifestyle. Diabetes, type 2. Diabetes often runs in families. Some genes affect how your body produces or uses insulin, a hormone that controls blood sugar. Obesity. While diet and activity matter, certain genes influence how your body stores fat or feels hunger. Cancer genes control how your cells grow and divide. Mutations in tumor suppressor genes, like P53 or oncogenes, which promote growth, can lead to cancer. It's important to remember, having a risky gene doesn't mean you'll get the disease. It just means you may need to be more aware and proactive. Personalized medicine. The future is now. What if your doctor could choose treatments based on your DNA? That's the idea behind personalized medicine, and it's already changing how we treat disease. Pharmacogenomics. This is the study of how genes affect your response to drugs. Some people break down medications too quickly or too slowly. Genetic tests can help doctors choose the right drug and dose. Example, people with a certain version of a liver enzyme gene might need lower doses of blood thinners to avoid bleeding. Targeted cancer therapies. Cancer treatments used to be one size fits all. Now doctors can test your tumor's DNA and match you with drugs that target its specific mutations. Example, Herceptin is used for breast cancer with HER2 gene amplification. Gene therapy, fixing the root cause. Instead of just treating symptoms, gene therapy aims to correct the faulty gene itself. How it works. Scientists design a healthy copy of the gene. They use a virus, modified to be harmless, to deliver it into your cells. The new gene starts working and improves the condition. Some successful examples: treating spinal muscular atrophy in infants, restoring vision in a rare inherited form of blindness. Gene therapy is still young, but holds promise for many diseases previously considered incurable. Lifestyle still matters. Here's something important to remember. Your genes are not your destiny. Lifestyle plays a huge role in whether a disease actually develops. Even with a genetic predisposition, you can lower your risk by eating a balanced diet, staying active, avoiding tobacco and excessive alcohol, managing stress. Think of it like this: your genes load the gun, but your environment pulls the trigger, or keeps it from firing. Ethical considerations. As we gain more power to read and even edit DNA, important ethical questions come up. Should insurance companies have access to your genetic information? Should parents be allowed to edit their child's genes? How do we make sure genetic medicine is available to everyone, not just the wealthy? These are big questions that society, scientists, and governments are still figuring out. Public understanding and input are essential. Part 6: Real-World Stories and the Frontiers of Genetics. Welcome to the final part of our journey, Part 6. So far, we've learned what DNA is, how it functions, how it shapes our health, and how it can be used in medicine. Now it's time to look at real-life stories where genetics has made a big impact and peek into the fascinating frontiers where the science is heading. Real-world story number 1: Angelina Jolie's preventive surgery. One of the most famous examples of genetics in action involves actress Angelina Jolie. She underwent genetic testing and found out she carried a mutation in her BRCA1 gene. This mutation dramatically increased her risk of breast and ovarian cancer. After consulting with doctors, she made the proactive decision to undergo a preventive double mastectomy, surgery to remove her breasts before cancer had a chance to develop. Her choice brought global awareness to genetic testing and preventive medicine. This story shows how knowing your genetic risk can save lives, especially when the information leads to early action. Real-world story number 2: the boy with bubble boy disease. In the past, children with a rare disorder called severe combined immunodeficiency, SCID, also known as bubble boy disease, had to live in sterile environments because they couldn't fight off infections. But gene therapy changed that. Doctors successfully inserted a healthy gene into the boy's immune cells. The result? His immune system started working. Today, he lives a normal life outside the bubble. This case highlights how gene therapy is no longer science fiction. It's real and it's working. Real-world story number 3: the Golden Rice Project. Let's switch gears from medicine to agriculture. In many parts of the world, vitamin A deficiency is a major cause of blindness and even death in children. Scientists engineered a type of rice called golden rice that contains beta-carotene, a source of vitamin A. It's a genetically modified crop designed to save lives. Golden rice shows how genetics can be used not just to treat disease, but to prevent it on a global scale. The CRISPR revolution. Now let's explore the biggest frontier in genetics today: CRISPR-Cas9. CRISPR is a powerful gene editing tool that acts like molecular scissors. It can cut out bad DNA, replace it with healthy DNA-modified genes with extreme precision. Scientists have already used CRISPR to cure genetic blood disorders like sickle cell anemia, make crops more resistant to drought, study brain development and neurological diseases. The possibilities are mind-blowing, but they also come with big responsibilities. Designer babies. Are we ready? One of the most controversial topics is the idea of designer babies, using gene editing to change traits before a baby is born. Could we eliminate inherited diseases? Maybe. Could we choose eye color, intelligence, or athletic ability? Possibly. But just because we can doesn't mean we should. What are the social consequences? Who decides what traits are acceptable? Will this lead to inequality between those who can afford genetic enhancements and those who can't? These are deep questions that scientists, ethicists, and the public must grapple with. The next frontier: synthetic biology. Another cutting-edge field is synthetic biology. It combines biology and engineering to design and build new life forms. Imagine Bacteria that clean up oil spills, yeast that produce medicine cells programmed like computers. It sounds futuristic, and it is, but it's also happening now in labs around the world. Synthetic biology could revolutionize medicine, energy, and environmental cleanup, but it also requires tight regulation and ethical oversight. Your role in the genetic future. You don't need to be a scientist to be part of this journey. Understanding genetics empowers you to make informed health decisions, participate in important ethical discussions, support responsible innovation. Here's how you can stay involved. Stay curious and keep learning. Talk to your doctor about genetic testing. If you have a family history of certain diseases, support science education and access to healthcare. Wrapping up, Part 6, the adventure continues. We've come a long way, from learning about DNA's double helix to seeing how proteins are made, to exploring how genetics impacts your health, to watching CRISPR reshape the future. DNA is not just a molecule, it's the story of life, written in code, shaped by chemistry, and brought to life by people like you who ask questions and seek understanding. Thank you for joining this deep dive into the chemistry of DNA and genetics. The science will keep evolving, and we'll be here to explore it with you. Stay curious, stay inspired, and remember, your body carries the blueprint of billions of years of evolution, right down to the tiniest molecule. Until next time, subscribe and like the video.